and that is all active challenge method, in which we have used 2 samples of each batch and different fractions of that. Overall, the potency of batch number 6 is an average is 207, and batch number 17 121.38 IU per dose. That is, however, as per the requirement of 40 IU of the WHO British Pharmacopoeia, all by challenge method assay. The purification was done. The UV adsorption at 280 require measurement of filtrate indicate that the toxoid has separated into 4 fractions. As indicated, batch-wise, the flocculation activity is recovered in 85 to 90% in first and second fractions. The antigenic purity of fraction 1 and 2 in all preparations were significantly higher than parent toxoid, whereas the antigenic purity of fraction 3rd were significantly poor. Differences in potency is observed between the original toxoid and combination fractions. In case of toxoid batch number 73, that is a parent low potency, the mean potency of fraction 2 were failing in fiducial limits as per WHO guidelines. However, when the combined in the fraction 1 of 76, high potency shows significant improvement in antigenicity. And in vitro assay, we have used one direct ELISA method to determine the potency. We also use the capture ELISA method. For that, we have used the sample at same concentration at 10 LF per ml. Active challenge method, which is the described by the British Pharmacopoeia and WHO. And if we will go for to test one batch of tetanus toxoid, we need 54 animals, mice or guinea pigs for a standard, and 54 for sample test, and 3 groups of mice, 18 mice in each group, and we also need about 40 for control mice. So a number of mice or guinea pigs required very high by active challenge method. These are the main batches of the bulk toxoid which we have done direct ELISA assay and active challenge method, and we found that direct ELISA assay which we have done to raise the polyclonal anti-sera against antibodies of batch number 73 as well as antibody against batch number 76. So these are the main results of geometric mean: 147 by antibodies against batch number 73, 169 against batch number 76, and active challenge method is showing 149 as potency. These are the results to comparative results of the capture ELISA as well as active challenge method. If you will see, almost the results are parallel by both the methods, either in active challenge method or capture ELISA methods. This is capture ELISA and active challenge method again for vaccine batches. Comparative results, this is of direct ELISA, capture ELISA and active challenge method. So if you will see the direct ELISA batch 105, 129, capture ELISA 146 and active challenge method 123. So we have done all about 1, 2, 3, 4, 5, 6 batches, and final geometric mean of direct ELISA was 147, 169, capture ELISA 186 and active challenge method 149. Direct ELISA was, now the main purpose is to, because the results are significantly, there is no difference in that. So the direct ELISA designed as an alternative for determining the potency of tetanus toxoid at bulk stage, because this is the same bulk can be used for 5 to 6 formulations of different combination vaccines. So if you have already determined the quality at bulk stage, then you assure that yes, this bulk can be used final formulation of all combination vaccines. Before formulations, you can find out the quality of the toxoid at bulk stage. Statistically, there was no significant variation in the potencies obtained by direct ELISA using tetanus antibodies, either batch number 73 or 76. Means that either you raise the sera against 73 or 76, there is no any significant difference in potency assays. There was good correlation between direct ELISA and active challenge method to estimate the antigenicity of tetanus vaccine. The result indicated that the direct ELISA could be used as an alternative to confirm the quality of toxoid at bulk stage before formulations. That is the main purpose for this study. The direct ELISA was found to be simple and quick to perform and require a small amount of materials. Also, the assay was found to be giving the reproducibility results of antigen levels. Capture ELISA also using monoclonal antibodies have been standardized and designed for assaying the antigenicity of toxoid bulk as well as tetanus component in DPT formulations vaccines. The potency results of DTP vaccine adsorb obtained were significantly lower than active challenge method. Statistically, there was no significant difference in the potencies of tetanus bulk obtained by active challenge method and capture ELISA method. It is more sensitive method, capture ELISA, and it is parallel to the active challenge method in which we are using many number of animals in either guinea pigs or mice. For tetanus bulk, average ratio of potencies obtained by capture ELISA / active challenge method was 1.24, whereas the ratio of DTP batches was 0.82. The potency of tetanus toxoid bulk by capture ELISA was 24.70% higher than ACM. However, it was 17.99% lower in DPT formulation, which may be due to partial desorption of tetanus antigen and interference by other antigen in ELISA. Because this is a final formulation, we have used diphtheria antigen also, Pertussis antigen also, aluminum phosphate gel also in final formula. So there may be some interference of the other antigen as well as gel also. Or it is not fully deadsorbed tetanus toxoid at the time of deadsorption. Now the comparative study of potency by tetanus active challenge method. Why we want to introduce the new in vitro method? Because this method is very cumbersome, cause severe distress. The one time the results of the tetanus vaccine you are getting very good, and other time the same batch you are getting the different results. So this results is very much variable in the terms of animal studies. Despite the use of large number of animals, more than 100 mice or guinea pigs to test one lot, the tests are not correlating with immunogenicity in humans. That is very important. If the tetanus vaccine potency is very high, and if you will go in the field, that is not correlated with the antibody level into the children or in person. That is already immunized with that. There is no correlation between the potency by active challenge method and correlation by antibody level into the human. Sorry. The US potency assay requires only 4 guinea pigs for immunization and 12 to 15 mice for titration per batch. That is in case of the antibody induction method. The titration could be performed by in vitro assays, thus saving further animals. Has an advantage over the active challenge method is that the usage undiluted vaccine for immunizations. Dilution of adjuvanted adsorbed vaccine may not reflect the true immunogenicity of the preparation which we are doing by active challenge method. Indian Pharmacopoeia also described antibody induction method as an alternative test to release the vaccine. It requires maximum 9 guinea pigs and 12 to 15 mice for assaying the potency of a lot. So the replacement of challenge method by serological determination of antibody titer in the immunized animals allow only a part reduction in number of animals required for that. In the present study, it has been tried to develop an approach by comparing active challenge method for testing potency of tetanus vaccine with proven antibody induction method. Antibody induction method we have used that Hartley strain of guinea pigs 250 to 350 weight, mice 17 to 22 gram used for antibody titration. Tetanus toxin we have used this batch number to determine the L+/100 dose of toxin, and standard tetanus antitoxin we have used as national reference standard of India. Now this is the antibody induction method. We got that 3 samples and the geometric mean value of 7 guinea pigs 7.7, 7.24 and 9.75, and average was is 8.2. However, as per the requirement of the Indian Pharmacopoeia, the potency by active challenge method should be 0.5 IU. However, we are getting approximately 8 IU. So there is no any correlation between the specific requirement by Indian Pharmacopoeia as well as WHO and British Pharmacopoeia. If you see, these are the batches which we have done 11 batches. Active challenge method potency is 140. against the requirement at 40 per dose, 80 per dose, and antitoxic unit 16.84, and we are getting the ratio 8.3, 10, 7, 10 like this, and average ratio we are getting 9.58. And antibody induction method against the requirement of 5 we are getting 15.05, very high. And the potency of active challenge method 144 against the 80 requirements. So if you compare by the active challenge method and antibody induction method, reference preparation, the ratio between, this is reference preparation which we have used for active challenge method assay. The active challenge method we are getting 80 requirement, this is the, it contains 80, mean potency 8.2 by active antibody induction method, and the ratio is 9.75. And in our batches, we also get the same type of ratio, tetanus vaccine 144, antibody induction 15, 9.58. Minimum requirement for active challenge method is 80, and active antibody induction method is 0.5. So against this 0.5 we are getting 15, against this 80 we are getting 44. So if you will see that there is no correlation by these methods against the requirements. These are the main findings. The ratio between IU and AU for TT vaccine was 9.58, which was very close to the ratio 9.75 obtained for reference preparation. On the basis of the result, it is postulated that it is ratio 10:1 is considered between IU and AU. The minimum requirement for potency of tetanus vaccine to give protection to human subject is 20 IU per dose in terms of challenge method. Antibody induction method, that is the meaning, because at present our requirement is 40 IU per dose, can be reduced at 20 IU per dose, which will also pass by the active antibody induction method. Antibody induction method gives in vivo results which correlate well with clinical protection, as is thus more reliable. During the present study, it is reconfirmed that the challenge method validity is variable each time the test is performed for LD50 value, challenge dose, fiducial limits, and statistical analysis. There can be occasions when challenge test needs to be repeated using large number of animals. However, in case of antibody induction methods, as less number of factors come into play which affect the final outcome of potency test results, and such a repetition is not required for that. Potency results obtained were found comparable with, did not show any statistical significant difference with those obtained by WHO recommended lethal challenge assay. For conventional antibody induction method, minimum requirement laid down in IP is too low, which may be fixed as at least 3 out of 9 Winnipeg sera and should contain more than 4 units of tetanus antitoxin. Antibody induction method is a simple, economical, and more reliable method which helps to practice 3R concepts. It is also possible to assign tetanus vaccine units of potency in terms of IU, even if antibody induction method is performed. Antibody induction method gives the true pictures to determine the protection afforded by vaccine against tetanus vaccine. Now these are the main direct method which we have used. Statistically, there was no significant difference in result obtained by direct ELISA method and active challenge method. The results indicated that direct ELISA could be used as an alternative, confirm the quality of toxoid at bulk stage. Capture ELISA also giving same type of results by this method. Antibody induction method, which I have shown the same type of results in that case. This is just, thank you very much. I have given these details with the Serum Institute of India sending our vaccines to in different countries. Thank you.
Thank you to Rakesh for an overall overview of the tetanus toxoid vaccine. So, are there questions?
Yes.
Yes, one.
So, depending on the assay, you're saying you have 25% up to 10-fold more antigen in terms of potency. So how do you translate that into formulation? And how are you going to predict protection in formulation?
I have done initially at the bulk stage of tetanus toxoid and final formulation of DPT group of vaccines. And both the methods, antibody induction method and challenge method, the main purpose to use the active challenge antibody induction method instead of active challenge method. So if we will go the correlation by 1 in 10, means if the potency is 1 by active challenge method, it can be 10 by the antibody induction method, like this. So there is a correlation can be used. You use only antibody induction method and find out what will be the potency expected by active challenge method without doing that test. This is the correlation which I have given to determine the potency by antibody induction method, antitoxic unit, and show how much potency we are expecting by active challenge method.